Hey, everybody. This is an intro video to two things. First, we're going to talk about metabarcoding, or in this case, fungal amplicon sequencing. So the idea is you would use common fungal primers, in this case, to amplify the ITS region, for example. And then you would do a second round amplification of those amplicons with barcoded primers, pool them, and then send them off to the sequencer without any need for library preps. The second part I'm going to talk about is our protocol called ISSR-seq. That stands for inter-simple sequence repeats. So basically what you'd be using is a single primer that is in and of itself just a microsatellite sequence, often with a few bases at the end as an anchor. And typically these are done in single primer reactions. And then they're run out on an agarose gel or something, and the bands are scored. But I'll talk a bit more about that in a second. What we're doing is taking this a step further and then doing a library prep on those amplicons and sequencing them. Okay. Share my screen here. Okay, so you can all see this. So we'll start off by talking about fungal amplicon sequencing. So before you begin, what you want to do is quantify your genomic DNAs, and you want them to be all roughly equal when you start out. That way that'll help you in terms of getting more similar concentrations by the end of this protocol, so you can sequence your samples. Okay, so quantify your genomic DNA samples with the qubit broad range double-stranded DNA kit. And then once you've done that, you'll know the concentrations of your samples, and you can dilute them down to 20 nanograms per microliter. If some of your samples are below that, then you can use extra input DNA to get up to about 20 nanograms in a reaction. Okay? And then in this case, prepare your primers at a concentration of 5 micromolar. So usually with a typical PCR reaction, we use 10 micromolars from 100 micromolar stock. So you dilute those 1 in 10. In this case, you'll dilute them 1 in 20. Okay, so you've got your DNA, your genomic DNA is roughly at 20 nanograms per microliter. You have your primers ready. Let's start with the PCR reaction here. And I'm going to play the video of Hannah, our graduate student, doing some fungal amplicons here. I'll just move this out of the way, move myself down here. How about that? Okay, so here's Hannah getting some fungal amplicons set up. And the way this is going to work is you're going to do this in 2 rounds. The first round is going to include primers that have tails on them. So you're going to have your primer sequence that matches the fungal ITS region, or the flanking regions of that. That's pretty standard, like you would do with Sanger sequencing, for example. But those primers are going to have tails on the other ends that allow you to do a second round PCR, right? So that's a common sequence that's outside of the more specific sequences. So here's the first round. Okay. You should expect targets from between 300 to 500 bases in length. So I think in this example, we're only doing one of the 2 internal transcribed spacer regions. So there's the ITS1 and the ITS2. We're only going to focus on one of them, because this allows us, when we do paired end sequencing, to actually bridge the gap rather than have just the bits on the end being sequenced. Okay. So when you set up your reactions, you're going to set up 25 microliter reactions, although you could cut this down to like 10 if you wanted to. We're going to start with 8.5 microliters of water, 12.5 microliters of PCR master mix, which includes the enzyme, buffers, DNTPs, etc. You could use any kit you want. I would suggest using a higher fidelity kit, so to cut down on PCR errors. And then you're going to use 1.5 microliters of your forward and reverse primers. Sorry, that should be reverse. Change that real quick. And then 1 microliter of your DNA, which is at 20 nanograms per microliter. Another interesting thing about the primers you'll be using is not only do they have tails on them, but they also have an equal combination of primers with an N in the middle, 2 N's in the middle, or 3 N's. And the reason for that is when you do something like amplicon sequencing on an Illumina sequencer, if you don't, if you didn't do something like this, all of the sequences would start out exactly the same, which could cause some problems downstream in terms of sequence complexity. So we do this to kind of randomize or jumble the I want to say reading frame, but it's not really reading frame, the start of the sequence, such that you don't bias the downstream complexity of the sequences. Okay, so you've set up your reaction, and now you're going to set your thermal cycler to the following conditions. So you have an initial melting time at 95 degrees Celsius for 3 minutes. Then you're going to do 15 cycles of the next 3 bits here. So 98 C for 20 seconds, 55 C, depending on what primers you're using. For ITS, we usually use 55 for 30 seconds, and then 72 for 30 seconds. This, if you've done PCR before, you'll recognize this is a pretty short extension time right here. And that's because we have a really small fragment of a few hundred base pairs. And then the last step, you're going to do a final extension of 5 minutes and then 4 degrees C to hold. So it's basically just a standard PCR reaction. We're doing 15 cycles here, which is pretty low if you've done PCR before. And that's because we don't want to do too much amplification at first. And so that can introduce biases in your analyses. For example, you can get chimeric sequences, and you can also get over-representation of certain specific sequences. So we want to keep this to a minimum, but get enough to where we have some PCR product to work with. So we start with 15 cycles. Okay, so that's our first round PCR. And now for our second round PCR. Okay. First, we have to do a cleanup. All right, so we have to clean up the products from that first PCR. We do this with Ampure XP beads. We use a different kind of beads called Oxygen. They're made by Corning, but they're virtually the same thing. And so, so you want to bring the beads up to room temperature and equilibrate them for about 20 minutes and vortex them vigorously to make sure they're all mixed into solution. And then you're going to use 32.5 microliters of beads. That's a 1.3x bead to sample ratio based on what your PCR volume was. And then you're going to pipette those up and down 5 times and let them equilibrate at room temperature for 5 to 10 minutes. You want to make sure you maximize binding of the DNA you just amplified to the beads. Okay, and then you're going to pellet beads on a magnetic plate, which you can see here. So this is an 8-strip tube. You could do this in single tubes, you can do it in 8-strip tubes, or you can do it in 96-well plates. Okay, but they'll form these pellets. And let me move Hannah's video out of the way here a little bit. That's not what I wanted to happen. Okay, you can see the pellets starting to form here. And basically, when you have a clear solution, it's ready for aspiration. So you're gonna take the liquid phase off and discard it, the supernatant. The DNA is bound to the beads on the magnet here. So you're gonna wash the beads with 150 microliters of fresh 80% ethanol. Remove the supernatant after about 30 seconds, and then do a second wash with ethanol. Then you're going to resuspend those beads in 22 microliters of TE buffer or water. Water works too. And then you can let them sit for another 2 minutes, put them back on the plate, and then the magnetic plate, and then take off the supernatant, which actually contains your DNA at this point. Okay, and so what you'll do now is proceed directly to the second round PCR. Okay. So before we do that, we need to quantify our DNA samples. That's because after the second round PCR, we want to be as close as possible to equal concentrations, okay? And so for this, we use the Qubit high sensitivity dsDNA kit. And then you can also run out your products on a 1% agarose gel. Okay, you should be able to see a little faint band, but you only did 15 cycles of PCR, so you might not even be able to see it, but that's okay. And then it comes time to set up your second PCR. So you've cleaned your first round amplicons, you've cubited them so you know their concentrations, and now it's time to move on to the second round. Okay, so here's the second round PCR. And this is going to include your barcoded primers. So unique 5' and 3' barcoded primers, which is going to allow you to separate your samples out after you sequence them. Okay, so set up your reactions in this order. It's going to be pretty much the same as the last round. You're going to use 7.25 microliters of water, 6.25 of your PCR master mix, 0.75 microliters of your individual barcoded primers, so the forward and the reverse primer, we call them I5 and I7. And then you're going to include 10 microliters of your DNA from the previous round. And you're going to do pretty much the same PCR cycle as you did last time, 15 cycles of these 3 steps, only your new annealing temperature is going to be 54 degrees C. And the important thing here is that based on the qubit results from your first round PCR, you can modify the number of cycles in the second PCR to get you an optimal amount of product. So if you have in the range of 1 nanogram, you'd use 22 cycles. So you're going to need more cycles to get up to a certain amount of DNA or product. 5 nanograms, you'd use 20, 10 nanograms, you'd use 14, 25, you'd use 12, and 50, you'd use 10. I wouldn't expect you to see 50 nanograms after only doing 15 initial rounds of PCR, but if that's how you would adjust your second round PCR. And so in that case, what you can do is you can actually take your first, let's say you do 96 samples in your first round. After you qubit them, you can bin them into categories. So that would be like, let's say if you had, everything under 10 and everything over 10, if you had a between 5 and 25 for all of them, you could bin them into two basic PCRs with different numbers of cycles. So you'd bin them so you can set up separate PCRs for those different concentrations, and that'll get you closer to an even amount in the end. Okay, and then you're going to do another cleanup. So this should say number 2 cleanup. You're going to do another cleanup. This time you're only going to use 20 microliters of your PCR product. Pretty much follow the same protocol as before. Leave that up there for a second. So if you want to pause it and look this over. And after you finish this, you're at a stopping point. So you can put your samples in the -20. Okay. And so then you would do another qubit for your samples, and you could run a gel and you should be able to see something this time. Okay. And then like we're going to talk about in the genomic DNA library prep video, you would then pool your samples at equimolar ratios. So you might have to dilute some or use some more of others, but you're going to pool them just like you would genomic DNA libraries and sequence them. That second PCR you did actually barcoded your samples on the 5' and 3' end. So you now have dual barcoded samples. You can pool them together, sequence them, and then sort them out informatically later. Okay, so the second part of this presentation, we're going to talk about ISSR amplicon sequencing. And this would be for if you want to do reduced representation of genomic DNA. So before you begin, just as before, you'd quantify your samples with the broad range qubit kit. You dilute your samples, in this case, to 20 nanograms per microliter. That seems to be a pretty good amount based on a lot of optimization we've done. So you dilute your samples to 20 nanograms per microliter. If they're lower than that, then you want to use more product. And then you'd prepare your primers in a standard way at 10 micromolar, basically, which is more or less the same with what you'd use with a standard PCR reaction. Okay, so you have two options as to how you could approach this. The first option would be to use a single primer amplicon, okay? Or to use a single primer in your PCR reactions. Okay, so you'd set up each set of reactions with just one primer. So you can just throw it right into the master mix and then aliquot it out, and just add your DNAs and go. The second option would be to include primers in multiplex. And so we've multiplexed up to 4 ISSR primers at once and gotten good results, at least on the gel. We haven't done any sequencing with multiplex. All we've done is single primer amplicons. And the main difference here is, like, if you use single primers, you do a bunch of separate reactions and then you pool them by sample. So you pool multiple primer amplicons by sample. So let's say if I used 4 different primer motifs in 4 separate reactions, I'd pool them together as a single set of amplicons and then take them forward. Whereas in multiplex, you just do this in a single reaction, right? So you'd use 4 or so different primers in a single reaction. And the idea here is that, you know, an ISSR primer is just a microsatellite, oftentimes with a little anchor on the end, like a YT or a TT or something, that allows that primer to bind at the inside end of the microsatellite motif. We also use unanchored primers occasionally, but this is a pretty safe way of doing things. You can also use multiplex amplicons, which would give you multiple amplicons, because not only do you have one motif that's matching at 2 different places, the forward and then the reverse complement, but then you have basically 4 times 4, so 16 different combinations of those primers. So you could potentially sample more of the genome by doing this. The thing you have to be careful of is the fact that you're using microsatellites as primers means that these repetitive sequences, if they have matching motifs between the primers, they can form really nasty dimers. And so you want to choose primers that are not really well complemented to one another. So you're going to have to pick these beforehand and make sure you're not using, like, you know, CGG and then GCC as motifs, which will stick together and probably won't result in very good amplification. Okay, so here's some examples of ISSR primers. They're just basically microsatellite sequences, and often you'll see a little anchor on the end here, like a TG or something. Okay, and I'm going to play my video of me setting up some ISSR amplicons here, just so you can see how it's done. It's just a standard PCR, so there's nothing really that fancy about it. So you'll set up your PCR either with single primers or multiplex primers. Here, what I'm suggesting you do, since you have basically 1 day to do this, is do a multiplex and just see how it looks. Okay, so here's your reagents. So we use roughly 5 microliters of Apex master mix, half a microliter of betaine, which is optional. You would just make that up with water otherwise. And then 1 microliter each of your primers, and then 1 to 1.5 microliters of DNA. Okay. Oops, let me skip down to our, and this is typically what an ISSR gel will look like. You see all these bands, sometimes they're present, sometimes they're absent. Traditionally, these were used for just scoring presence-absence characters as dominant markers. So, you know, you create a matrix here where you have your different markers on the x-axis and then your different samples on the y-axis, and you'd score 0 for absent, present for 1, 1 for present. There are some obvious limitations to using dominant markers. They don't really give you much information on heterozygosity, for example, so variation within an individual. And so what we're doing is using these banding patterns, these amplicons, and then just doing a library prep on them and sequencing them, which turns them from dominant markers to co-dominant markers. So now you have information on, if you're dealing with diploids, potentially heterozygous individuals, right? So you have 2 copies of the genome, you're going to get SNPs that could be heterozygous, right? So now you're using co-dominant markers. Okay, so here's the basic reaction setup as I was showing you before. we rounded up a little bit in the video, but this is roughly the amounts of the different materials you would use. I like to use betaine. I think it helps a little bit with specificity, but maybe it's just one of those magic things that you put in your PCR reactions and hope for the best. I think it does help a little bit, but it's optional. And then here's the thermocycler conditions. So we do an initial melt with 4 minutes at 95, 30 seconds at 94, and then we do 30 cycles of 45 seconds at 50 degrees Celsius. You can go lower, but that might mean less specific binding. You can go higher, which might mean fewer loci. That's something you probably want to optimize ahead of time and play around with this a little bit. Do that for 30 cycles, and then we have the extension time here is 2 minutes, and I'll show you why here in a second, with a final extension time of 10 minutes and then 4 degrees C for hold. If you're setting up multiplex reactions, you would just make up that lost volume by not adding any water. So in this case, we're adding roughly 1 microliter of each primer. So you're gonna have 4 times the amount of primer that you would in a single primer reaction. And so you just add no water to make up the difference. And the conditions would be exactly the same for the thermocycler. So I think we should focus on this one for the workshop. So try to add each primer and then just don't add any PCR water here. Okay, and so what do these amplicons look like? Well, this is an example from Japanese stiltgrass that I did. I think it's Japanese stiltgrass. On a 1% gel, I used 4 primers from the set I showed you earlier. And what you can see are the first sample, I think, might be a genomic DNA sample, that smear. But you'll see a number of distinct bands and then some kind of smearing going on. And what I think that is multiple bands that you just can't distinguish from each other. Okay, so each one of these you would carry into a library prep. Okay. So if you're using single primer individual reactions, then I would suggest after you've run the gels for each one of those, you pool your amplicons from each one of those single primer reactions into a single individual. So if you used 4 primers, you'd take 4 times 3 each, and you'd have 12 microliters. You could use more or less if you wanted to, depending on how much you need for your libraries. If you're multiplexing, then you can just proceed immediately below. You don't need to pool anything because the primers were already multiplexed. So you should perform bead cleanups on the PCR products based on the protocol we showed you earlier. And then use qubit high sensitivity double-stranded DNA assay to figure out the concentrations of your samples. And what we do is when we take these forward into standard library preps, we dilute the samples to 5 nanograms per microliter. That seems to have worked out really well for us. We've tried all kinds of permutations here, and this one seems to work really well. And then you would just proceed with a standard SPARQ library prep. Okay, so that's going to be what we cover tomorrow. And I'll leave it up to you whether you want to try genomic DNA or your ISSR amplicons if they look good today, as to whether you want to do preps on those or do preps on the genomic DNA. The only difference is going to be the fragmentation time. And so with ISSR amplicons, we use 3.5 minutes for a fragmentase time. That seems to be the optimal amount of time for products in the range that we see here. I'm just going to go back to this gel here. We see products from about 200 bases up to several kb, like up to 5 kb in some cases. And the nice thing is the bead cleanup actually gets rid of the very smallest amplicons. It also gets rid of any primer dimers or leftover primer, obviously, that might be left over. So it cuts off anything below, say, like 200 bases or so. So that's really useful in that sense. Okay, so that's the end of the ISSR presentation. So you just take these amplicons right into library preps, which is what we're going to cover tomorrow. All right. Thank you.